Okay, good morning. Let's get it started. So today we are going to first look at graph and network in MATLAB to see what the function support for graph and network analysis using built-in function in MATLAB. And then next, we will look at comparative network analysis. For single cell data, from single cell data, okay. So this use my single cell gene regulatory network construction and then compare using the toolbox I developed. 3, we will see how can you try the single cell 24-fold analysis. So this is a machine learning pipeline we build for single cell data, and you can compare between 2 samples. And you can also do virtual knockout. So this includes network comparison. Let's see, called single cell gene regulatory network construction and comparison. And also virtual knockout. Virtual knockout. So you know what it means, the knockout, that basically you delete a gene in a system and to see what's the phenotype change caused by this gene knockout, and then you can infer the function of the gene. We will do this using established network and then get rid of one of the genes and see what's the impact of this deletion of the gene to the network, so we can predict the function of this gene. So it's data driven, and you can delete many genes if you like. So I will show you how to do this easily. And then number 4, I will let you spend 5 to 10 minutes to do the course evaluation. So we do have this online AEFIS. So this is the website. Last, we will still look at the spatial transcriptome analysis. So here we are going to use a geo dataset as an example. So real dataset, we will see how can we download a dataset from a geo database, including the image. Then we do alignment between your measured spots of expression, then align to the image of section, and then to do the spatial analysis for this. Okay. Now let's go to MATLAB we are going to try to look at graph and the network in MATLAB. Okay, so first, if I gave you a network, okay, if I gave you a network like this, 1, 1, 2, 3, 4, 5, and this is linked. I use exactly the same example in here. So this is wrong. We have a triangle here. So if you have a network like this, can you create the matrix representation for it? So let's try this. If you're not looking at the PowerPoint, can you just type this matrix in MATLAB
You take out?
for this graph? So let's call this adjacent matrix, which
Yeah.
so now your task right now is to look at the topology of this network. You have 5 nodes, and manually type its adjacent matrix into MATLAB. So we have adjacent matrix for this network. Okay, in the adjacent matrix, 0 means these 2 nodes are not connected. 1 means they're connected. Adjacent matrix should be a squared matrix. In our case, we have 5 nodes, so this adjacent matrix should be 5 by 5. A node itself, for example, 1 to 1 itself should not be linked in this case because we have not shown the self loop between a node between the self. So in the diagonal position from a top left to the right bottom, you should have 0 in the diagonal position of this matrix. If you finished, please paste your source code to the chat so I can copy. Okay, so this is our adjacent matrix. Okay, so as long as you have this definition, they are ready to make a graph in MATLAB. So let's call this called graph. Okay, so this graph, we'll take that in. Now we have a graph object. So graph is a specific data type, also called class in MATLAB. It's self-defined, but it's already predefined by MATLAB itself. But you can create your own classes yourself. So this class is called graph, and also if you look at its properties, you have a table for the edges, a table for the nodes. And the nodes, you can put the name of nodes, use strings or character arrays to label the nodes. But without giving any name, the nodes will be just labeled as 1, 2, 3, 5 to 5, okay? The edge right now is a symmetric matrix, just defined in A, and it's unweighted, so the connectors are just ones. If you like, you can take a look. I double click this and it tells you 1, 2 linked, 2, 3 linked, and the weight as 0 as 1. Okay, so that's a definition of the object. There's no node number ID, so just leave it blank. So what you can do with this, once you have this object G in there, you can certainly just look at what the property already in there. Okay. The first thing you want to do is to plot it, right? So you can do the plot. And you want to assign this plot into a P variable, because you may want to change the plot later. Okay, so that's your graph. Okay, because you already assigned the output of this graph plot into a P variable, so you can have a P dot and look at its properties. Say, okay, I want to change color, edge colors, for example, change to red. And you can do this very easily, okay, by assign a new values to the properties you have found. I think the most interesting one is to change the layout. If you do this, I select, you have different several layout method you want to look at it. And if you do a circle, all the nodes will be arranged in a circle. But here we only have 5, so it's not very clear. It's a circle. It's a pentagon, basically, right now. But we will use more complicated examples to show the usefulness of this. Okay. Okay, now we are going to create a more complicated network. So this one is unweighted and also undirected. Now we are going to create more complicated, and I just use a random number generator. Okay, I'm going to do a random number generators here. I'm going to create a 20 by 20 network. Okay, now this adjacent matrix is fully connected because every node from 1 to 2, 1 to 20, they all failed. So this will create a very busy network. I don't want to do this, so I'm going to do some kind of filtering. So I require a greater than 0.7 to be kept. So I only by chance leave 30% of original links in the matrix. If it's greater than this, that will be true. So let's see, I will keep that. Then my matrix B will be only contains those greater than. Let me see if I can do this. This is not a good idea. Let's see. Yeah, so this B become vector. So what I should do is to do a dot product. Okay, so I just multiplicate. If it's a 1, I keep 1. If it's 0, becomes 0. So now my B matrix become a filtered version of A. So only have those linkers stronger than 0.7 be kept. So this is kind of cut off. Now I can create a graph for B. And this time, you create a graph, but you cannot use graph. You have to direct it. So DI graph of B. Then this G become a directed graph. Okay. Then you can do the same, just as we described. You can plot this into this figure and also keep the plot as a variable P. And then you can change. Also, you notice there's some of this self-link. So do you see the loop? Because the 3 to 3 is not 0, because here, and you have a link self loop. And you see the direction, so from one node to another, okay? So now you can try this layout, and this is a force, there's a circle, then you have a different presentation of the graph. Yeah, just give it a try, and I will let you spend 3 minutes to play with these functions. By the way, any questions so far? So I'm not a salesperson for MATLAB or MathWorks sales representative here, but I just found it convenient. Of course, if you like use R or Python to achieve all this, you can. Nothing wrong with other languages. Many languages have its own packages. Mm-hmm. Yeah. Yeah. It's not. So if you do, if you stopped here, for example, and you tap this, and you push tabs, the tab key, it'll give you suggestions of all this. Yeah. Okay, so what else we can do with this? What kind of question you will have if you are given a network like this? So in the lecture, I talked about the centrality. So you probably want to have some computation on the centrality of all these nodes. So I show you how to do this. So let's see centrality, some kind of centrality is this, and we have a centrality, the function. This function will take a graph, and then you type the types. Okay, so again, the tab key will have a suggestion of all this possible centrality you can compute. Because this is a directed, so you have a so-called closeness centrality, but consider inward links and outward links separately. And the page rank is Google, the algorithm for Google page, web pages. Between centrality, we didn't mention it, but it's commonly used. The definition is, if you look at one of the nodes, you will see, okay, all the other nodes, if you have a pairwise shortest distance, how many of the pairwise short distance will pass the node you are looking at? The more shortest path between all other nodes will have to pass this particular node, then this node will have a high between centrality. So this is a very useful technique to measure the importance of the nodes. So here, I just gave a C between the centrality to a variable C. So this C is the variable 20 by 1, so this vector. And let's see which node have a largest C values here. So if I do max C, and also I will get the index. I don't care which one is the largest, what's the value of the largest. It could be 30, it could be 25, 26, but I care about which node has the largest. Number 10 has the largest node. So number 10 is here, okay. Another question is which one is the second largest? So how can you do this? You use max k and see, okay, give me top two. So the index will be 10 and the next one. So this is a function in MATLAB. So to give you 10 and the number 4. So these are the two top so we have this highlight function for the P and maybe index. I'm not so sure whether we can highlight anything, but yes, so this highlight function and the P is the picture of this plot. And I gave the index and highlight these two nodes. Use a slightly larger circle. If you compute the C using not between centrality but the page rank, you got a different C, and then you do the same to find the top two index. So for the page ranker, the top two are number 1 and 14. So slightly different. Number 1 is more important based on the page ranker. I like this. I don't know if you can do color. Okay, so just you see you using different measurement of centrality, you have a different view of the importance of the nodes in the network. Next, we can try is to find the shortest path from one node to another. For example, if I try to see, okay, how can I move from number 9 to number 7? Maybe not linked directly. From number 9 to number 12. Okay, so I want to look for the shortest path from number 9 to number 12. Consider the weight between the nodes. So I'm going to parse. We need a graph. We need number 9 to number 12. Okay. So this is one of the algorithm implemented in this function called the shortest path. It takes the graph itself, and then you have a starting node and destination, the target nodes. And then this parse will be computed. If you look at the PS, the parse PH, PH contains 4 values, 9, 10, 14, 12. So this is parse. So from 9 to 10 to 14, to 12. So that's the parse it found out. We can highlight this parse by putting this ph here. How can we do this? Okay, so I just highlight this edge color with red. So you see from number 9 to 10 to 14 to 12. This is the shortest parse we can search and found out by using the algorithm. So you can give any pairs of the combinations. So from 3 to 20, for example, you can do the same thing. If you do all the pairwise search, and you will see a majority of them, the most will pass through number 10. Because number 10 was found to have a highest between centrality. What that means, most shortest parse will pass it. And number 4 is the second largest between centrality values associated with number 4. Any questions? Okay, fine. Now let's do a documentation reading. Doc centrality. Well, Yeah, if you're on this page, I want you to open this so we can go through this example. It has enriched representation of a real roadmap network. Okay, what you can do is if you're on this page, you can see, okay, click live script. In this case, you don't have to type anything. You can just click the button, say run the section. So you highlight this section or double click this blue vertical bar here, and then you just run this section, load example data, and here you have X, Y, their coordinates. Okay. I just demonstrate this by click on this. Yeah, I'd rather to pop it up so I can move the window. So this is a network already have all the information and where the location of the nodes. The coordinates have been given. Let's show this. In this case, you compute the closeness centrality and also label the okay, so here the closeness centrality was computed for this network, and the value has been translated into the color code, and its node was labeled with highlighted with its color by its according to its closeness centrality score. Okay, so those cities or stops is in the center of this whole map. So their value and the closeness centrality is higher. That means if you travel, start from this center and to all other nodes, then on average you should have a shortest distance in the middle from middle. Of course, it's the case. Okay, so the next one is to consider the weighted edges. The first one was not considering just any link, just have a weight equal to 1. But here it's consider the weight between each links. Then you get a slightly different outcome. In this case, we compute the centrality unweighted version of a betweenness centrality. Okay, so those ones, most important ones. Okay. Okay, this is done. We are moving to the second topic. We will look at how can we construct network using single cell data. So in this case, we need to bring up the toolbox. So I'm going to close this in case just assume you don't have any questions. Any comments or questions? Let's just clean up this. I will do close all and clear all. If you have a single cell gene expression analysis toolbox installed, you should be able to do CD, GEA, and change your working folder to here. If you don't have that, then you have to Google and search how to install it. So what I will do is to do GA toolbox Bing search, and you do have a documentation and a quick installation. Just in case you don't have this installed yet, so you can copy this and just download and zip to your working folder, basically. If you have this, let's try GA2. We are going to use example data. Okay, so now we have this example data have 3080 850 genes. So you can construct a network for this transcriptome level gene interaction. So that will be a network between 3,850 genes by 3,850 genes. So that will take some time. So that's why this menu here. So see, okay, single cell 24-net construction. Basically, that will construct a transcriptome level of gene regulatory network. And you see the two turtles in there, so don't click. It will take some time. Okay, take at least 30 minutes or longer. However, we have a button here. Okay, so here you can you select a subset of genes. Instead of using all the genes, you can select, I'm only interested in 20 genes here. So I can select these 20 genes and create a local network. Still be fine. So you have options to select those genes you want to include it in your network. And also you can say you paste the name of genes in here, as long as the name of genes is in the list. Here we just randomly have those genes selected. As a demonstration purpose, you can just select this and click OK. And this will create a network for you. So this is a network for this 20 gene. Okay. The method we use to create this network is called the PC regression. And that's what we found is one of the best way to do this, considering not only pairwise correlation between genes, but also consider all other genes to be have influence on these correlations. So we summarize all other genes impact use PCA and incorporate that effects into pairwise comparison between coefficients. And here you get a directed network and also it's weighted. So the blue indicate this positive correlation and the red indicate negative correlation between these two genes among all the cells you're considered here. Okay. So so this network right now is representation of all genes expression in all these different cell types. So this is not a really good good way to do it. So we should really extract part of this cell type. Say I only work on the B cell or T cell, create a network that will make more sense. But now it's a mixtured cell type. So this just for illustration purpose. What you should do is to extract the cells. Okay. For example, only monocytes, and you create cell type specific gene regulatory network for each of them separately. Okay, so once you have this network, you can change the size of the labels. Okay, you can change the weight of these links. Let's click this. Okay, circle it around this until you think it's reasonable. Everything is proportional, so and you can change the layout. We have some options of different layouts. Okay. Okay, so what you see here is a directed network, and you can average the links between two genes and make it unidirectional. So if you click this, it will change a directed graph into undirected. Okay. Because most of the time, if you have negative correlation between A and B, A negative correlate B, B will be also negative correlated with A. So both of these directed to linkers and the weight should be negative. So the average will be still negative. So you still keep the same color and their relationship, they're very similar. And the next, you want to remove some of this weak link so that you can use this scissors to see, okay, I want to keep 70%. If you see, okay, 95%, then you will show only 5% of age, the top ones. But here is, let's say, okay, 70%, 65%, and you will remove those weak link and only keep the majority of links. Then you should try some of the layout again and give you the better presentation. And also you notice some of these nodes already highlighted in red. Those are the non-transcribing factors in the database. So we have a database and their transcription factors then will be highlighted as a red. Assume they're more important in the network as a regulator. Okay, this button show you animation and to show you the purging of the edges by its weight. And you start from remove the weak edges until you remove all of them, so you can have a dynamic view of the trimming. You can stop anywhere, and if you want, okay, the density or the dense of this network is already lower enough, I will stop it, and then the picture will maintain like that. Okay. Do that again. No, I just randomly select 20 genes for demonstration purpose. Yeah. And the other thing that takes the super long time is average all the genes. That'll tell you, like, when you do this stuff, that'll really tell you, like, the most important stuff. Yeah, for that one, if you have a node more than 200, for example, you only want to show in the graph because you don't see them when the network become huge. So in that case, you only use statistics to compute those index and see which one is more important than another. But for small network, you can visualize the change. I know, like, yeah. 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 It's more focused. And we have a study, it's conducted by Eric, and he tried create a transcriptional level of the network and also create this local network like 20 genes. The relative weight you create will not change, the consistent. So it's okay if you only select 20 genes, you focus on that, your result will be very similar when you consider everything first, then zoom in to the 20 genes you're interested in. So
is there a way for certain pathways
yeah.
affected?
Yeah, you can. If you know the name of the genes, you can have the options. So for example, you know them, you paste in there, or you selected by alphabetic. So you know, okay, these genes are in the same pathway. You just put in the CD14. Then you can create a network for those genes. Yeah. Okay, so you may still have questions, but I want you now to jump to number 4. And so we roughly have quick demonstration of this, but now we will take a break, and also please help me to finish this. I will send, I'm not so sure you're familiar with this. Looks like this is a system that allow you to give the feedback. So I'm going to paste this page to the chat. Whether it should be in here or not, I don't know. So maybe it should be this one. But you may know better than I. Let's take 10 minutes break. So you finish, and also take a break and come back to the 11:05. All right. And also the course evaluation is more important, probably more important than you thought, and to us as instructor. Okay. Okay, let's come back to this main interface here. And you see, now you know how to construct a gene regulatory network from single cell data. As I said, it's best to do it to create a cell type specific networks. How can you do this? So you have to say, I will select the cell by class or by types. So here you use cell type, and you select one of the cell type for B cells, for example. Then select, then you extract those cells out, and then from this subset of cells, you create a network. Okay. So this network is for B cell specific the network will be very different from the one in other cell types. Let's try if I do a list of this. I copy the gene list, so I know next time I will do the same for another cell type. So now I create this for B cell, and then I extract cell type for monocytes. In this window, I create network again. I paste the same gene set. I just copied from the previous B cell, and then these two networks, one is for B cell, another for monocytes. So you see the difference between them, right? Looks like this ID1 gene have a lot of negative correlation with other genes in monocytes, but not here. And this MAF1, a lot of negative correlation with others in B cell, but not in the monocyte. So in this way, you have a cell type specific single cell gene regulatory network. Okay, so if you want to do this small network comparison, and you can also see, okay, I want to compare to single cell gene regulatory networks. So this button here is just construct one network, but this one will construct two for different subset of cells. So as what I did for B cell and the monocytes, you can select a grouping variable and then select B cell compared to with monocytes. So this two, and then you paste the list of genes, and then you, rather than independently do these two cell types, now I just using this interface to have a side-by-side view of these two networks constructed from a B cell and the monocytes. And also, if you have two batches of data, control and the disease, you can select that as a separate variable, and you select the genes and the cell types maybe to make this comparison. Also, you can also still have this image animation cut off to trim off both sides of these links. Okay, at a certain stage, you can stop. Okay, I think you think a signal is strong enough, you just keep that, and then you can use different layout. Okay, so this layout will put the same gene name at exactly the same position to let you to visualize the difference directly. Okay, make this font right, make the weight. Yeah. So what we can see from here is SCO2 gene, and have a lot of negative correlation with these members here, but not in monocyte. And the negative correlation between these two is really strong, but not here at all. But again, those genes are randomly selected, so maybe not make any sense, and maybe there's some new biology we don't know. But in your research, you may have a focused subset of genes. You want to look at those genes, share the same pathway, or you know what you are looking for, and you can include those genes in there. And maybe at that stage, you have better interpretation about this difference, but it depends on your user case. I have no idea what you will discover from your data. Okay, the transcription factor is called STAT. STAT4 here. Okay, so this one. Okay, any questions? So we did this. Okay, compare. Now I'm going to show you this one here. Okay. So this is a global level, the whole transcriptome comparison, rather than just focus on 20 or small set of genes. Again, that will take very long. I'm not going to click any of these buttons, but I just show you where the comment is. So here you have, this will help you to construct two networks and then compare. These two networks are transcriptome level, so that's thousands of genes by thousands of genes that you merge and compare using the manifold alignment. That won't be very helpful because this will just take 12 hours or 24 hours. You just leave this desktop running, you should get the answer. It's more complicated. If you send your data to the cluster, you have to know how to call the MATLAB function in there. You have to set that up. Yeah, it won't change much. You still have to wait, say 12 hours. Yeah, but locally it should be okay. So this option is more systemic. So you will construct de novo two networks, and they will compare, depends on your batch IDs, but normally you will compare one sample against another. You don't want to compare the cell types, but you have the options here. As long as you know how to group your cells into two groups. So normally I suggest you will extract the same cell type. So you focus on T cell and T cell. And then T cell you have, from patients, you have control, the two groups. In this case, you will have options. See, I want to have a batch ID compare. And your basic cell population is T cell, so you don't care about the cell type. You just compare these two clear population, but only differed by their condition. Then you create a network independently for these two data sets, two batch of data cells, and then align them and find the most different part. Okay, and if you do this way, and you will have the parameters. At the end, I will still have the warning. So this analysis may take several hours. If you really continue, you will log your computers here and for 24 hours. And it's difficult to break it down. So I will click no. But that's the way you, if you decide to go that far for your data. Okay. If you have a set of cells, again, I encourage you to have a pure cell type. So here I want to have T cells. So I want to study if I knock out the gene from T cells, what will happen? So this will be the link of this virtual knockout, a single gene. Okay, my background, my starting point of the population of cell is pure T cells. I don't want to mix up with all other T cells. So the prediction will not make any sense if you have a mixed population of cells. Your knockout is impacted to B cell or T cell, we don't know. But here we have clear pure T cells, and you can do virtual knockout. So I will select, I will construct the network, I will select the gene. So for example, we will knock out this gene from systems. It will construct the network and delete that gene from the network and see which other genes will be influenced. Okay. I hope you are done with this. Do you need more time? Or any questions about the network analysis using the data here? Single cell gene regulatory network. Yeah, I hope everything is okay with you. So let's see anything else I missed for this tool here. So you know you can change the type of this visualization of cells. You can change the colors, right? You know that. You can that's close all the other windows. You have a switch between 2D or 3D. I just click this because this is for publication purpose. That's embedding the delete cells. You know, you can extract the cell into individual population by cell type, for example. Okay, this show. Okay, this is T cell. This is monocyte. This NK cells. We have. Okay, basically most of them. Okay, if you don't have any other questions, we are going to move to the last topic. Remember, this is a new topic. If you took this course last year, we won't have this available because at that time the data is not available, the tool is not available, or the methods or the algorithm hasn't been developed. So that's only happened just in the last 12 months. Okay, so now I'm going to show you the latest development of this related tools. And again, we are now in the documentation. I suggest we switch to the download, the folder. You want to download the package to here. Let's see, you and Dave. Remember this, we still do the same thing, then you will be ready to go. So I will type this, unzip into chat. So that's what you need to download the package and unzip it to the current folder. So I suggest the current folder, working folder should be the downloads folder. I'm going to do the same thing here. So let me know once you're finished. Okay, in my case, it took me about 1.5 minutes. Now I should have this folder. I will double click. I'm in this folder right now, so I'm ready. Okay, so next I'm going to go to the geo database. NCBI geo database. Now I'm going to search Visium. So you know this is a platform by 10x Genomics to do this transcriptome analysis platform. If you click search, you got 170 results. So some of the results will contain both expression level of your spots and also the image. So we will take a look and see what sample, what tissue, what species, some of those basic information. So let's see, for example, here, so this a neuron, and I just open this link and look this record here. So this one sample, and this one Yeah, everything is zipped, so it's not easy to handle. So it may work. You have to download and zip the folder, the files in there, but I'm not going to use this. Let's see this one, integrated analysis of this. I'm looking for an example data set so I can directly import from the web link. Again, they save this into zipped file, so I have to unzip it. And in the sample, they only provide this low resolution image. Maybe this will work. So I'm inside one of the samples here. Yeah, I don't know, but we can give it a try. So if it will work. So if you see some samples from is this from Visium? Yes, Planiform. It's supposed to be a Visium sample, and you see, okay, you have a expression matrix, you have an image, then this could be a sample that we can import. So I will copy this ID, the sample ID. Okay, all I need is this ID, GSM 5772311. Okay, I copied this. It may work, may not, but let's try. So I'm inside this package, this folder here, so I can have access. I have this access to this tool here. And this, instead of SCGA2, so now ST. ST stands for spatial transcriptome. You select the geo accession number here, and then you can paste that number. And you can use those example numbers if you like, but here I'm going to show a random sample I pick. I never seen it, but hopefully I can get it wrong. So here I type in the sample ID with the Visium technology already have the spatial transcriptome data and image. So I type this ID in here and let the program to go there to try to find, okay, we need a position of the spot. So that sample is not complete, so we cannot use it. They didn't upload the position example of the space. So I have to move to another example.
I'm getting an error that says that its STGA tool is not being recognized.
Yeah, you should. Have you seen this?
Yeah, I'm in the folder. I also think I changed my directory to this folder with the CD command, and it's still not. I don't even see the tool command on the left though. Yeah, I don't see it there.
Do you have this?
No.
No, then you're probably in the wrong folder, and or you haven't download the latest version. So you make sure you have this unzipped, download fresh.
Yeah, I do have. I can try it. I'll just try it again.
You at least you have to see there the function should be there. Has to be there. Okay, in this example, Mhm.
And I'll tell you like the kind of files that are. Results.
Recent results in this web browser. The one we just used.
Yeah, whatever search you did
Mhm.
on
Oh,
API.
yeah.
Yeah, if you go to the video tab, you'll click like download data.
Yes. So you certainly, if you can download the data, unzip it. Yeah. Then if you have that, you may want to use, see, okay, we have this to look at the folder. But what I want to show you is directly download from for demonstration purpose. I mean, just found the right sample, the complete record of this, so we can just do this by just one click. Okay, so here I have a sample with matrix data, image, and this position. Maybe this will work. So now my target sample is this. Okay, so I paste on the chat, in the chat, see which one I'm testing right now. I'm not sure it will work or not, but just try. Not if it's complete, I should be able to download this. So now it's the ID I'm trying now. This will go to this web page to download all the samples. Okay, we get something in there. Okay, we have a slide of tissue, and we have expression matrix on this tissue, and then you can click this button. So I want to view the 3D. Okay, if you can get this, let me know, and we are ready to move to next stage. Are you ready to move on? Okay, first, you see we need to resize our data points. So here, you need to, so we are doing alignment. So we'll align this dots and perfectly with the tissue. So we will click this button to show all this alignment related. So I will first do a zoom out, because now the image section is too small and my data points just too big. I will zoom out a little bit. So this zoom out is all direction. Okay, so make it comparable first. And then I want to zoom out by the y-axis because it's too long. So I make it smaller, this direction. Okay, now they're almost comparable. I'm going to change it later to make a further adjustment. Now I can click this movement. Now it's highlighted. I can start to move my data points to align them. Okay, run to this. By the way, I don't think we are right now, because if you look at this bubble here, this bubble empty spot here, and this one is down. So we actually need to flip or rotate my my coordinates of this. What do you think? So yes, we need a flip. I will do the flip first and then rotate. So I will flip by along the x-axis, along y, I don't know. Yeah, it's okay. I will flip it and then rotate. Let me rotate it first. Yeah. No, so I still need a flip it over. And then I move. Still not right. How should I do it? Not the right shape. I really don't know how to align this. This is a special case. Mostly, it's very easy. Just do one flip and then they're very clear structure. You can align them. Flip by again. This circle should be here. Yeah, so I think that's the correct way. What do you think? This sample is really weird. I mean, normally I will look at the bubbles here and to match those. Well, also the shape, for example, this shape here is a curve, should be here actually. But this alignment is important because one of the way to do this classification for these dots is based on what's the picture under it. So I will do an image analysis by looking at each point underneath what's the image, the small pixel looks like, and then classify them. So the alignment is important, but in this case, for some reason, I just don't know how to align it.
I think the
Mhm.
line with that kind of goal as on the back of
Far left.
on the bottom there.
Yeah.
That
Yeah.
is like.
Is
the
this one?
border
Is
that-
this one here?
Yeah,
Okay.
those two that is sticking up is not the more obvious.
Okay. So you think this one? Okay, I flip by x axis here. Yeah. So that's the shape line. This one here. Okay.
Yeah.
Okay. Good. Okay.
It's just a little from the top, like it doesn't actually line up with the top of it.
Okay. 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 I just assume this is correct. But I guarantee it's not so hard for most of the samples, not like this. We can make it smaller so it fit perfectly and it's guarantee and zoom along the x-axis, whatever. So at the end, you will achieve some of this, okay? So I just assume this is a perfect alignment already. I can rotate by a certain angle if you need it, but I found it's mostly you just need to have a rotation 90 degree. You can hide any of this picture by clicking this my button and look at the original. So we do have a bubble here, maybe. Then we want to remove some lowly expressed gene because now we have 31,000 genes. That's quite a lot. And I only keep this, like gene has to be expressed in 55% of all the spots. Now I keep 10,000 genes left. So you can look at the gene expression. Say I want to select multiple genes randomly and look at their expression. So this on the picture. And this is very uniform sample. So not a very clear structure in there. So I can just show some of these gene expression patterns. So you can click this button to close all of them. Okay. Yeah. So you can use SC image to use expression matrix to classify all the points. But here I'm going to show you example data instead of this because you have to pre-compute those. So I shut this down and then just use our example data. So processed. So you see this is aligned very easily and you can rotate. You never mess up whether this aligned or not. This one is perfect aligned. In this case, I already pre-compute the cluster. So this is based on the gene expression profile. And the green one, all the dots have a similar expression profiles. So the group has a number 4 or number 5. And then this a yellow one on the edges here, and it's classified as a different group. So you can see that underneath this image have a strong structure compared to the one I just used as an example. But this one is more heterogeneous sample, and there's different clusters. And then another way is to base on the image. So this clustering algorithm just look at the picture under each of this spot and group them by the texture of the image, by the color of the samples here. So that's a slightly different organization of the clustering. Then you can search, say, oh, I need genes with highly variably expressed across those sections. Then you identify those genes have a feature, for example, highly expressed in certain part of the slide here. So the top 10 genes. So you see some of genes highly expressed in this particular region can be identified. Okay. Yeah, and also you can so once your alignment is finished, you don't have to do that again. So you just switch off this alignment tool and just save your results. And next time you load it back, it's still perfectly aligned. So you only need to do the alignment once, manual alignment. Then you can change the size of these bubbles, change whether you use a circle or the field. You can change the color. Function hasn't developed. 2D, 3D, select genes to express, to look at the expression. Okay, random genes, for example, to look at individual genes expression. Color code in based on different clustering algorithm. So SC3 gave you slightly different clusters for the cells. I shouldn't see their cells, they are just spots, okay? One spot maybe have a multiple cells in there. And you can select, let me see why it's something wrong with this. Yeah, you can highlight part of this region and select the spot and found the highly variable genes for highly expressed gene for this spot. So you get some idea what they are, and looks like there's a lot of collagen related genes. So I would like to think this part is some tenders, maybe, I don't know exactly. A sample, a tissue with a lot of collagen in there. Yeah, so those are the options you can have. Also, if you click this button and you come back to the single cell expression analysis tool, so you can manipulate all the spots as a cell. You can do the embedding, and for example, to do the t-SNE rather than the location, and then you use t-SNE to do the clustering, and then you can match back to their position. Anyway, so that's the demonstration of this tool at this current stage. Yeah, so I haven't got the time to show you this classroom. It's based on the image. Yeah, but that's basically, that's all. Hopefully, yeah, so I needed to install some of this package to do this image analysis in this particular machine before I can run this clustering algorithm. But anyway, that's all I want to share with you until today, and I will see you next Tuesday for your presentation. I hope everybody will have a very successful discussion about your project, real data or hypothetic, whatever. But remember, we need to have significant innovation and your performance and your approach, the reasonable approach, and your performance, the delivery. So 4 categories of the criteria to judge whether your performance is good or not. And the homework will take 1/3 of the score, and the presentation 1/3, and the final exam will be 1/3. So that's the proportion. We will mix all your individual section of scores and summarize into a final scores for your final C. It's whether A or B or C, or maybe it's 85 above A, or 75 to 85 is B, something like that later. Okay. All right. If you don't have any other questions, we will see each other next Tuesday. Thank you. Bye-bye.